Greetings and welcome to the third and final installment for the online cardiac lab. This is part 3, talking about cardiac output. Just a little bit of a note here. We are going to be seeing some terms and some things that we won't necessarily cover in the regular lecture. Nevertheless, those terms may be relevant to for exam purposes. Just terminology per se, like in diastolic volume and that sort of thing. So this is the most clinical of the three, so trying to transition us into a more clinical understanding based on what we know from parts 1 and 2 and obviously lecture. So good luck and thank you, and I'll see you later.
We have all felt our heart beating more quickly or more slowly, depending on our activity. Let's learn more about how heart function changes to meet metabolic needs, whether hanging with friends, walking to class, or running to catch a bus. Cardiac output is the volume of blood pumped out by each ventricle in 1 minute. Cardiac output is the product of heart rate times stroke volume, averaging 5 liters per minute at rest, whether lying down, sitting, or standing. Heart rate is the number of beats in 1 minute, averaging 75 per minute at rest. Stroke volume is the blood ejected by one ventricle with each beat, averaging 70 milliliters per beat at rest. The key factor controlling heart rate is the influence of parasympathetic and sympathetic stimulation. Parasympathetic stimulation decreases heart rate, while sympathetic stimulation increases it. We maintain a resting heart rate of 75 beats per minute because at rest, parasympathetic input is greater than sympathetic input. Removing parasympathetic stimulation allows the heart rate to rebound to the intrinsic rate of the SA node, around 100 beats per minute. Further sympathetic stimulation is required to achieve a maximum heart rate of 190 beats per minute. Changes in cardiac output also occur by altering stroke volume. Let's examine how this occurs in the left ventricle. When diastole ends, the ventricle has filled with blood. This volume of blood is the end diastolic volume, or EDV, shown here as 120 milliliters. Ventricular contraction ejects blood into the aorta. The amount of blood remaining in the ventricle after contraction is the end systolic volume, or ESV, shown here as 50 milliliters. Stroke volume, the amount of blood ejected during contraction, is the difference between EDV and ESV. In this case, stroke volume is 70 milliliters. These changes in ventricular volume can be shown graphically on the monitor. As blood enters the ventricle, the volume increases to a maximum value called the EDV. This high value on the graph represents the EDV. As the ventricle contracts and ejects blood, the volume decreases to a minimum value called the ESV. This low value on the graph represents the ESV. The difference between EDV and ESV is the amount of blood ejected, called the stroke volume. There are 3 factors that affect stroke volume: preload, contractility, and afterload. The first factor we will examine is preload. Preload is the amount of ventricular muscle stretch caused by the end diastolic volume. The greater the EDV, the more the muscle stretches, increasing preload. Stretching muscle fibers toward their optimal length allows them to contract more forcefully while returning to the same ESV. EDV, and therefore preload, is affected by changes in venous return, the rate of blood entering the heart. Venous return varies with body position, sympathetic activity, and skeletal muscle contraction. Increasing venous return increases preload. Slowing the heart rate can also affect preload by increasing ventricular filling time. In either case, increasing venous return or ventricular filling time increases preload. This is shown as an EDV of 140 milliliters on the monitor. This increased preload results in a greater stroke volume of 90 milliliters being ejected. The second factor affecting stroke volume is contractility. Contractility is the force of contraction at any given EDV. Increased sympathetic activity increases contractility, so more blood is ejected with each contraction. This results in a lower ESV at the end of contraction. Consequently, an enhanced stroke volume is ejected with the same preload. The third factor affecting stroke volume is afterload. In order for the ventricle to eject blood, it has to generate enough pressure to open the semilunar valve. Afterload is the aortic pressure that the left ventricle must overcome to eject blood. The pressure is indicated here as 80 millimeters of mercury. Hypertension causes aortic pressure to increase, raising the pressure necessary to open the semilunar valve. The increased afterload is indicated here as 100 millimeters of mercury. With increased afterload, more of the force of contraction is used to open the semilunar valve. Consequently, less of the force is available to eject blood. Therefore, an increased afterload reduces stroke volume. As you have learned, increases in preload and contractility increase stroke volume, whereas increases in afterload decrease stroke volume. Let's review what you've learned. Cardiac output is the product of heart rate times stroke volume. Sympathetic activity increases heart rate, while parasympathetic activity decreases it. Stroke volume increases with greater preload and enhanced contractility. However, increased afterload decreases stroke volume. These factors allow cardiac output to vary dramatically to meet the body's metabolic needs.